Y'all say hello. See, then tell your mom that you're on YouTube. I can turn the camera around, I guess. All right, so what we've been watching, keeping up, we've been discussing enzymes and enzyme mechanics, and this is a critical part of our overall foundational discussions because the mechanisms by which protein structure influence protein function is critical. And what we've been looking at is ways of thinking about how we can carry out metabolic reactions using proteins, using amino acid polymers that act as catalysts, enzymes. And we have a pretty good sort of analogy and definition about how we can break things down, and we can think about how we can build things up using the addition of a phosphate, which isn't always the way we carry out anabolic reactions, but often it is. We have been then going through some of the enzyme mechanics that are sort of classical enzymology. And what we have then is the idea of doing a rate experiment in which we keep the enzyme concentration constant and you change the amount of substrate. And just as the definition, substrate is the stuff that the enzyme is acting on. And if we do an experiment like that where we keep the enzyme constant but we run a series of rate assays looking at different substrate concentrations, we find that is a canonical classic enzymatic phenomenon of what we call the standard enzyme rate look. And that is, just for those of you that are nerds, a logarithmic function. An exponential function is what you would see like that for a growth curve. But enzyme rates typically are logarithmic functions. And so whenever we see something that is got a rate equation like this, and we're talking about substrate concentration, we must always recognize, that is an enzyme catalyzed reaction. And I just say this, when we start talking about the pumping mechanisms for moving glucose into a cell, we'll see that kind of rate equation. And one of the things that we can kind of conceptually think about this is that given a fixed number of enzymes, there's going to come a point in which there is so much substrate that every enzyme has its active site filled at any one moment. And so adding more substrate doesn't make any difference because the enzyme is working as fast as it can. That's Bmax. Now, what we can then do is somewhat artificial, but it is an interesting way of comparing, is this notion of Km, which is the substrate concentration at the more linear portion of the curve at 1/2 Vmax. And Km is a cool idea about a way of thinking about the protein-substrate interaction. And we can actually apply Km to pretty much any kind of protein interaction that you can think of. So the way a protein might bind DNA is going to have a Km. And changing the amino acid sequence, changing the structure, and you can do that by changing conditions or changing the amino acid sequence, is going to affect the Km. So when you think about, from a genetics perspective, you have a dominant mutation, a mutation that is working, that causes a protein to do something more. What we've done is affected the way that enzyme, in an enzymatic reaction, is interacting with its substrate such that we're maybe lowering the Km slightly. It has a higher affinity. So this way of thinking about the enzymatics is useful. Now, we have then the definitions of competitive and non-competitive inhibitors. And this is another sort of interesting function of the way we set the whole experiment up. And that with a non-competitive inhibitor, what we're essentially doing is making it so the population of enzymes is not working as effectively. One of the enzymes may be bound some kind of inhibitor that is blocking its active site, or it's binding to the top of the, some aspect of the enzyme and changing the shape, twisting the protein around so that it can't work anymore. And with a non-competitive inhibitor, if you have removed effectively the number of enzymes in the population, then the overall rate is going to be decreased. And no matter how much substrate you put in, because you have fewer enzymes, the maximum velocity is never achieved. And we then, sometimes I think this is somewhat artificial, but we can then say that by doing the math, doing the graphical analysis, that the Km is actually been pushed further down the x-axis. The Km is larger. Now, for a non-competitive, or for a competitive inhibitor, what we're doing is probably, my way of thinking about it, is we're affecting the substrate in some way. And now you've added like a synthetic substrate or some kind of non-hydrolyzable substrate, and you can get around this by simply having more substrate. You can compete it out. And so the substrate concentration is going to be higher to achieve Vmax, but Vmax is achievable. And then because of the way the calculations work out, we say that the Km is not being affected. So that's an interesting thing to think about. Now, enzyme regulation is this whole other thing, and we'll touch on it a little bit in a couple of spots, but I'm not going to spend too much time on that. But there are a number of ways enzymes can be regulated, and primarily they're going to be regulated, in a couple of ways. You either have the enzyme not present, just don't have it, and then you have some kind of inducing agent that's going to turn the gene on, and the gene gets transcribed, the RNA is translated, the enzyme is present, we're off to the races. And this might suppose then, in order to shut the enzyme off, you have conditions such that proteins don't have a very long half-life. And so if you shut the enzyme production off, the enzyme concentration drops, and you go back down to a ground state. Bacteria tend to do this more than we do, I think. It's fair to say. As we'll start seeing as we get into transcription and translation, the production of an amino acid polymer is metabolically pretty darn expensive. And if you don't need a protein, maybe you just better keep the information tucked away as DNA, not transcribe it, not translate it, don't produce the protein until you need it. The problem with that is, if you need it right away, well, sorry, you're out of luck. It's going to take me a little while to build that protein. Now, bacteria are pretty fast. And so they can go from essentially being silent to having the enzyme present in like 10 or 15 minutes. You and I pretty much require probably 3 or 4 hours. So if you decide you need an enzyme now, you're not going to have any significant amount of enzyme before lunch. So if it's something you need right away, you better have it present and then just have it in a state that it's not active. So we can have a variety of different kinds of systems then that will either induce or inhibit an enzyme that is actually in the cytoplasm. So we got a lot of enzymes that are floating around in our cytoplasm that are not particularly functional right now. I just don't need to have that metabolic pathway going on, or I don't need to have that kind of cell signal going on. And so we can activate proteins. We'll see as we go through this, a standard way of activating proteins is to add a kinase or add a phosphate to them. That'll activate a lot of stuff. You also, for catabolic enzymes, might have some kind of molecule that's binding to the enzyme and acting as an allosteric inhibitor. Just to throw that term out. This means it's changing the state, it's changing the shape. So an allosteric inhibitor. And this would be like a small secondary protein, or it could be even metabolite. A really cool mechanism is what's known as a feedback mechanism, in which the product can act on the enzyme, and once the product builds up to a high enough concentration, it is going to bind to the enzyme and block it from continuing on, kind of like a thermostat. And again, we have the idea of Km here, because you might have an enzyme inhibitor type feedback system. So we'll just think about you're breaking something down and generating a product. And the Km that the enzyme has for that product is going to influence how much of the product you generate and how active the enzyme is. So an enzyme that has a really low Km for its inhibitor is going to be really sensitive to the production of this product. An enzyme that's got a fairly high Km is going to produce a fair amount of product before it finally has enough concentration to bind. And so you can change the sensitivity of enzymes. I often think a little bit about a thermostat as a feedback mechanism. In that, you all have thermostats in your home. We run it at 68 degrees. You turn it down to 64 at night. You go to bed, you come back up, turn it back up. Now, what you probably don't ever play around with or sit there and stare at is that thermostats have what's called an offset, in that it's not very good for the system to run, shut off for 3 minutes and then turn back on again. So, like with your house, you run the heat for a while, and then it hits a set point, it shuts off, and then the offset might be 2 degrees, and you actually, with a digital thermometer or a thermostat, you can set that. And what you're waiting for is the house temperature to come back down below the set point, and so if it's set at 68, it might actually get down to 66 before the thermostat kicks back on. It'll run, and it won't go to 68, it'll actually go a little bit above that. And so, you have this kind of wave function. And the closer the offset is, the tighter you're maintaining that. For some metabolic systems, we want to have a very precise amount of this material floating around. And so you got an enzyme that's got that kind of control. For other systems, it's like, let's produce a bunch and then shut down, and we can go way down and we'll produce a lot when it turns back on. So different kinds of feedback mechanisms exist, all sorts of things happen, and enzymatics is a fairly complicated area, and it becomes an interesting area for drug discovery because often your drugs are going to be inhibiting different kinds of enzymes, and when you inhibit them, did you slam it shut so it don't ever turn back on again? Maybe not the best drug. Does it have a structure that cross-reacts and binds with other proteins, and so you end up with a variety of side effects? So it becomes actually an interesting issue for pharmacology. So there are a lot of different mechanisms, and we'll be covering some of the feedback mechanisms and things as we go. Now, the other aspect of this is that because the enzymes are working based on their secondary and tertiary structure, ergo, they are critical. The primary structure is critical to the enzyme function. If we change the primary structure of a gene product, of an enzyme, we might change its Km a little bit. And this actually is what's responsible when we start thinking about all the differences we see within the population. Because your cheekbones grow at a certain rate based on a protein's ability to bind a DNA sequence and cause a gene expression to either be cranked up higher or lower. And very slight differences in that protein's structure are going to have slight differences on how much of a specific gene product might get turned on and how fast your cheekbones grow. So, it's one of the things that we think about. I don't know if you've ever seen the movie Gattaca. We all put that on the list, where you can take a drop of blood, sequence the genome from the white blood cells, not the red blood cells, in humans don't have nuclei, get the sequence, and it's probably no more than, I bet, under 100 genes that are really critical for determining how your face is going to look. And with sufficient algorithms, you can take that gene sequence and say, okay, you've got a G at this spot. That means you're going to have a phenylalanine at this spot in this amino acid. That's going to subtly change the structure, and you're going to have this kind of nose or this kind of look to the spread of your eyes. And using these 100 different genes and the way they may interact, we really should be able to come up with what a person looks like at adulthood. And so there actually had been, there's a pretty cool artist that was found bits of DNA in New York City, like everything from a piece of chewing gum on the ground to hair. And she was actually only looking at like maybe a dozen genes, if that, and had a fairly primitive algorithm that you could 3D print a face based on the genetic sequence. And it's not like a particularly exact match, but it's sort of like, oh, that could be your brother, that could be your sister. I mean, it's pretty cool. And then with sufficiently complicated algorithms, we really should be able to get you a picture of this is what your kid's going to look like at 25. You know, you're not going to necessarily be able to say whether they're going to have facial hair or whether they're going to decide to have a nose job, although there may be some psychological aspects to that we don't even know. But genomic analysis for these kinds of traits is a pretty cool trick. Additionally, one of the things we're finding out are the different kinds of gene sequences that affect the face and the possibility of modifying them. So you can say, I really want my child to have George Clooney's jaw, and my nose is actually too small. I need to have a bigger nose. What are the gene sequences we need to manipulate to actually change that? Because I don't look like my Uncle Herman because I stood next to him when I was a child. I look like my Uncle Herman because we share genetic sequences. Just some things to think about. You don't know my Uncle Herman, though, so it's all right. All right, so we then have talked a little bit about these kinds of rate equations in which we're looking at different kinds of environmental conditions. What is the cytoplasmic environment that the enzyme is in, or the protein is in? And we have the whole notion then of the optimum structure for a protein's function. And then we've got suboptimal spots. Different proteins will have different kinds of optimum conditions. And you know, you would like to think that everything in the human cell is going to have a pH optimum of 7.2 and a salt concentration of 0.9%, and it's going to be at 37 degrees centigrade because that's what we always are. And it just isn't that way. I mean, because enzymes actually have a fairly wide range in which they can work. You know, so you can characterize enzymes based on different environmental factors and see what the optimum is. And again, changing individual amino acids can change the optimum. So you might have something that works better at a slightly higher pH or a slightly higher salt concentration. So that's how we can begin thinking about genetics and protein function because of this very cool shape change stuff. Let's see. That catches us pretty well down the road for chapter 3, I think. I didn't bring my book, and I should have. And I got my set of figures, but I just don't feel like throwing them out yet. And so I'm just sort of thinking about anything fun to talk about. What the real old versions of this textbook used to do is at this spot, they went through the initial steps of glycolysis, the breakdown of glucose, as a sort of an example of an enzymatic pathway. And it really doesn't work very well here because you're only telling a little part of the story. It's like, yeah, whatever. It's just an enzymatic pathway. And so we now have a whole chapter dedicated to protein structure, which we've been talking about all along anyway. And so that's what chapter 4 is getting into, is the idea of protein structure. And I thought what we would do is kind of cover chapter 4 a little bit about further discussion of protein structure, like blah, 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 protein structure, I get it, but also some of the techniques involved in doing protein analysis. And some of these are techniques that we will be kind of calling back on later in the semester, and it's worth talking a little bit about. These are some of the techniques we're going to be doing, are things we'll be doing in lab very near the end of the semester, but it's worth talking about. So, if we think about proteins, the way we're going to work with them biochemically is not a one-size-fits-all sort of model. And I will tell you that my PhD was focused on purifying proteins from fruit flies, proteins that were involved in cell adhesion and cell-cell rearrangement, which is a really cool phenomenon that happens. And we were purifying proteins from fly culture cells, from fly cells. Purifying proteins is the kind of thing where every single protein that you're going to be working on is a little bit different and has different characteristics, sometimes dramatically different characteristics. The great advantage of shifting from working on a protein to working on the gene for the protein is that DNA is always the same. There are great big, huge recipe books for DNA manipulation because you can isolate DNA from a bacterium or from a giraffe and you work with it the exact same way. And that is a really nice thing. You don't have to figure out every single protocol for DNA. It's already done for us. For every single protein, they're always a little bit different. And just as an example, the idea of protein solubility. Some proteins are very water soluble. Some proteins require a very specific salt concentration. Some proteins are just flat ass not very soluble. Like a protein that's stuck in a membrane is going to have a really big hydrophobic stretch, and if you take it out of the membrane, it is not going to be in solution anymore. And so oftentimes we have to use detergents. Detergents, as we've been talking about this, have got, they're amphipathic, and so they got the nonpolar stretch and the polar stretch. They're able to interact with proteins and cause them to be water soluble. Now, a side effect of this is very often the detergent is going to destroy the secondary and tertiary structure, and so we say it is in fact a denatured protein, and it may very well be non-functional. And that would suck if you've got some kind of enzyme assay, but the enzyme itself is stuck in the membrane, and in order to get it out, you have to denature it, either work on a different protein, or you got to figure out a different kind of system that allows you to analyze this. So solubility plays a big role when you're thinking about manipulating proteins. Another thing that is critical is overall charge. It can be very negatively charged. It can be very positively charged. It can be nonpolar. And this plays a significant role in how a protein can be separated and isolated. So, let's just decide that we've got some sort of interesting enzyme assay or enzyme we want to work with, or a protein that we want to study. What are some of the things you can do? First off, you got to figure out a way to grow up cells or tissue that have that protein, and that can be a real pain. Consequently, a lot of the early protein biochemistry was done on proteins that are incredibly common. And so a great protein to work on is the albumin, which is a generalized term for a very soluble protein that's found in blood serum. And you can get a lot of blood from cows, and consequently bovine serum albumin is a protein that's really easy to purify, and it doesn't do much, but you can do all kinds of analyses with it. Other proteins that are extremely rare may take a lot of material to extract a small amount of protein. Again, another reason why the ability to do molecular genetics has a tremendous advantage, because you might have a protein that is present only on a few cells and only a few copies of it. So purifying the protein might be really, really difficult. But if you can find the gene for it, you can pop it into bacteria and you can produce buttloads of it. So that's a way you can begin twisting things around and kind of using molecular biology to your advantage. Doesn't always work that way, but so now, let's say though we want to purify a protein. We want to get it to a single protein band. And there are a number of different techniques that are useful that we'll be talking a little bit about. First, you might be able to purify it based on its solubility. You know, if you can extract it and say there's a reagent you use like 5% or a 2% saline solution, and that doesn't extract a whole lot else from the tissue you're working with, you're really lucky. Generally that doesn't happen. But you get a protein mix and you solubilize it. There are a couple of tools we can use that we can separate out proteins. One of the tools we use is something known as gel filtration. And this is a form of column chromatography. A gel filtration column is often like 3 or 4 feet tall. And it is a series of beads that are smaller than a grain of sand. And you have this column packed with beads. The beads have little holes in them, kind of like little tunnels passing through them. And gel filtration separates proteins out based on size. Smaller proteins and smaller molecules end up getting caught in the gel filtration matrix, and they are taking a much longer path. The big molecules can't get into these little holes, and so they're they're coming straight out, whereas a small guy might be doing. And so, based on size, in that smaller molecules is slower, which, depending on how your perspective is, we'll see there's other systems in which smaller molecules have the advantage and they're speedier, but in gel filtration, smaller is slower. Another technique, now gel filtration is a fairly crude way of separating out proteins. It works, but it's not great. And you won't end up with a single protein. You're going to end up with a collection of proteins that are about the same size. Another thing you can then do is something that is known as ion exchange chromatography. And this is based not on size, but now on charge. So what you can do is have some kind of like a positively charged bead, and just as a general note, most proteins, at least in animals, are going to be negatively charged, going to carry a net negative charge. That's just the nature of proteins on our planet. And so they'll interact with a positively charged matrix. By increasing the salt or by changing the pH, you can begin to knock stuff off such that the things that are weakly charged, they can get knocked off first. Things that are carrying a really big charge, they'll stick to this stuff a lot longer and require a lot higher salt concentration or a lot more of a pH change. And so you can begin knocking things off based on charge. So you can begin to combine these guys where you can get a size fraction and then a charge fraction, and hopefully at that point you're getting down to just a few proteins in solution. It's the kind of thing that my dramatic pauses get a little bit too dramatic here. You can put it down on paper and have a brilliant protein purification procedure, and then actually putting it into practice can be like a massive pain. It's a big business though, because so many different kinds of modern pharmaceuticals are somehow being generated in cells, and you can produce insulin in a bacterial cell line, but you really don't want to inject that entire protein lysate into some dude and say, oh, here's your insulin. No, that will not be good. You need to purify it. And so guys at Tate & Lyle and ADM, man, downstream purification is a big, big deal. So this is two of the ways you might do it. Now, happily, because downstream purification is like, it could be like a showstopper. Every one of these steps, you end up diluting stuff. It can be just a real mess. A great thing to do is something that's known as affinity purification or affinity chromatography, in which you've got a matrix in this column, whatever, that excuse me, has got some kind of little molecule sticking off of the bead of silica that is going to bind your protein. And when it does, you can take a crude lysate, pass it over an affinity column, bind only your protein, and then by using either a little bit of a pH change or a little bit of a salt change, you can slightly deform the protein such that it'll let go and fall through. Affinity chromatography is a brilliant trick because now you don't have to go through all these steps. You simply take your lysate, that is, blast the cells apart, pass it over an affinity column, wash it a bunch to make sure all the contaminating things are removed, and then you do whatever it takes to elute the protein, and bada bing, you can end up with a pure solution, and that is a great trick. And just as a little plug, one of the things that we often will do in molecular biology is clone a protein. So you have a cloned gene, in which, by that meaning, we take the gene from whatever organism, from the elephant, put that DNA sequence into bacteria, and then have the bacteria begin expressing it. And a fairly simple trick that we actually can do upstairs is we can add an affinity purification tag. As it turns out, if you take the amino acid histidine, and as I was saying on Wednesday, you certainly don't need to memorize your amino acids, but it's probably not a bad idea to familiarize yourself with the list of the 20 amino acids. Just so when someone says, oh, there's a serine there, but it was a phenylalanine, or talk about histidine, you go, oh, that's an amino acid. Next semester in biochemistry, you can memorize it, but for us, you don't need to. Anyway, if you take histidine and you have a stretch of 6 of them, it forms, when you do actually a 3D model, it looks like a knuckle. It looks like a little ball. And you can use that knuckle in that this actually will bind nickel ions. And you can then do a nickel ion affinity chromatography using what's known as a 6-his tag. And that is a really cute trick because now you can force an affinity purification tag and you're off the races. Another thing that is done that actually gets into some overall protein structure stuff is the idea of making an antibody. And I would imagine that talking about vaccines and antibodies is something that people are probably a lot more familiar with now than they were, say, a year ago. And the different kinds of vaccines, and we'll actually talk about some of these different vaccines as we get through the class. So a standard way to make a vaccine would be to take a protein from the virus, whatever you got, and you generate a pure protein from that virus by taking a piece of its DNA, putting it into bacteria and making a whole bunch of it, putting a 6-his tag on it, purifying it over a nickel affinity column. And this is a column, in my lab, we run nickel affinity columns, and columns that are about that big. In an industrial pharmaceutical place, they're running a nickel affinity column in something that looks like a 50-gallon drum. I mean, it's like, a whole different world. But you can then purify large amounts of a specific protein and then inject that. Antibodies are themselves proteins that bind to something that is known as, in the generic term, as an antigen. And this actually has been an area in which the mechanics of enzymes has actually been elucidated, been discovered. So you have an antibody, and they're made up of 4 proteins, 2 identical light chains and 2 identical heavy chains. And the stuff that you're going to bind binds up here. So antibodies have the ability to bind 2 molecules, and as I said, the vocabulary, this is referred to as the antigen. So when you're vaccinated, you're being given the antigen. Now, of course, there are a bunch of different vaccines that are rolling out for COVID. The first 2, Moderna and Pfizer, these are RNA-based vaccines, which is a pretty cool trick that was developed back in the 90s, but this is the first time it's been rolled out in a really big way. And what we're doing here is injecting somebody with an RNA sequence for the major spike protein, and the cells that take that RNA up, they translate it, put it up to the cell surface, and it looks like they've been infected with the virus, but of course they just had the spike protein. Your immune system recognizes it and makes antibodies against it, and you are now you've developed an immune response. So that's a real modern way of doing it. A very modern, but not quite that modern, way to do this would be to take a portion of the virus, say like the spike protein or the envelope protein, clone that into bacteria, and you put a little 6 tag on it, and you purify the protein, and now you inject the protein into folks. That works, and that's actually been a kind of vaccine technology. The AstraZeneca and a couple of other guys, they actually are using either the virus that has been autoclaved, heat-killed, that's a standard way of doing the flu virus, or in the case, I think, of AstraZeneca, they actually are putting the spike protein into a non-pathogenic adenovirus and infecting you with this. And so you have this virus floating around for a little while, producing the spike protein, but not producing any of the different kinds of deleterious effects that the actual virus has. So it doesn't bind to cells in the lungs and things. And so it floats around for a while, and after a few weeks, it's actually going to be completely gone from your system. And it's probably made in such a way that it can't replicate on its own, so you can't cough out the vaccine, which would be actually a sort of interesting way of spreading it. And so vaccine technology is all about proteins and antibodies, and we'll talk more about them as we continue on. One of the cool things about an RNA-based vaccine is that it's very easy to shift the RNA to change along with the different variants of the virus. I mean, it's like really easy to do. Whereas if you're trying to actually make the cloned gene, you got to step back, make a new bacterium, and start the whole process up again. And it is a good bit more difficult to do. So an RNA vaccine actually has the ability to shift around pretty fast. Let's see. All right, so antibodies actually take us into a little bit of a spot where we can begin to look at how enzymes actually might be working. And I'm kind of like taking a little bit of a side break here to talk about enzyme function, just because it's a good place to put it in. So there actually was a company that was called Abzyme that developed an antibody system that could mimic an enzyme. And it works like this. Particularly for a catabolic, breaking apart enzyme, we have this idea that the way enzymes work, enzymes have their highest affinity for the substrate in what's called the transition state. And the transition state, just in a simple way, we can think of it like this, where we've got our molecule of A, B, and it's got a bond we got to break. So in order to break that bond, we probably have to expose it to water, because it's going to be a hydrolysis reaction, and it probably helps to torque it a bit, because when the bond breaks, similar to when the bow breaks, but different, when the bond breaks, it's going through a state that is called a transition, where it goes from here to something that's going to look like that. And then finally, pop. Or snap, or crackle, whatever your favorite sound would be for a bond breaking. This spot is the transition state. And antibodies that are enzymes that are breaking down a bond, they have a real high affinity for that. Now, the way this would work, because enzymes are stabilizing or lowering the energy of activation, what they're doing is they're stabilizing and kind of holding molecules at this spot. So we can think of this again sort of as our heat analogy. As you add heat to a solution, it starts getting more and more bendy, and eventually it snaps. Well, even at non-hot temperatures of 37 degrees, there's always a little bit of flexing going on. And what an enzyme is doing is shifting the population of molecules to this transition state and making it easier for things to break apart. So just, a way of thinking about this is maybe 90% of the time, the bond that we're trying to break is in this position. And then every once in a while, it pulls apart, or it shifts. So it's kind of very slowly flexing, or it's hanging and then it flexes and then hangs. And an enzyme that is going to bind here is going to shift the sort of population, if you will, to not being 90% straight, but maybe 90% held like that for a few moments, and cause an increase in the rate at which things break apart. I often think about it, if I'm a protein, sit down, you heard this already. I got 2 minutes, man. If I'm a protein and I don't want water molecules to punch me in the stomach, I keep my hands always here. And I'm walking down the street, and we have water molecules, I think of it like little 6-year-olds, trying to punch me in the stomach, but they can't. And then every once in a while, though, I check to make sure that my wallet and cell phone are still there. But most of the time, I'm keeping my hands over my stomach. What would happen then if there is an enzyme that when I do this, they grab my hands behind my back, and now all of a sudden, all these little 6-year-olds punch me in the stomach and I throw up? That's a way we can think about the transition state, is that if that is the reaction we're trying to catalyze, that's the transition state. Most of the time I'm here, I can stay at this, but an enzyme will shift the equilibrium in such a way that, oh no! And so that's how we can think about this. What the company did, Abzyme, is they built an antibody against the transition state. What you have to come up with something they call a transition state analog, which it turns out to be is really, really hard to find. But it is a chemical that has, as a stable structure, something that looks like the transition state of the reaction you're trying to catalyze. And there's not a whole lot of molecules that do that, but they found, they had an example. And the antibody binds the transition state, and it acts as an enzyme. So that is one of the ways we can think about protein structure, but what I had done was step out of my protein technology discussion to throw that in, because I had forgotten to throw that in at the beginning of the class. Hey, I can edit the tape, but I'm not gonna. On Monday, we will continue talking about protein technologies and talk a little bit about gel electrophoresis and things like that. That's all I know. Have a lovely weekend. It's going to be a beautiful weekend. It's going to be like, I think it's going to be maybe in the 70s even. No. It's going to be cloudy, and it's going to be butt cold, single digits. Just think about all the creatures, though, that are living outside and that don't have the ability to maintain their own body temperature. They're not doing much. Hey, I'm going to kill this. And I will see you cats